Hey everyone, so today I'm going to be talking about ATP synthesis and the biosynthetic pathways that we've learned thus far in our labs. So I hope everyone can see. It's kind of blurry, but this is the only way I could get everything on one screen. So initially in our first lab here, we have our pentose phosphate pathway, and that helps to generate some of our intermediates in our other pathways like glycolysis. So just keep that in mind. So our pentose phosphate pathway is going to start with glucose 6-phosphate, which is generally derived from like glucose in our diet. That's actually going to be one of our starting intermediates in the glycolysis pathway. So glucose that's derived from our diet is going to be broken down into glucose 6-phosphate, and that actually can come from the pentose phosphate pathway. So we can restart this cycle with that. So glycolysis is going to be the process of breaking down our glucose into pyruvate. So up here we can also see that we can get our glucose molecules from other things in our diets like lactose and galactose, and galactose can be broken down into our glucose 6-phosphate as well. So that's where we start with glycolysis. So with glycolysis, we're going to actually have to add ATP and able to get energy from this glucose source. So it takes 2 ATP to form 4 ATP in glycolysis. So at the end, we net a pyruvate and 2 ATP. So then we can go into the pyruvate dehydrogenase complex, and our pyruvate is going to come from there. And then so that pyruvate is going to get dehydrogenated, and we're actually going to get out some energy molecules as well in the form of FADH2 and NADH. And we remember that those can have a conversion to ATP. NADH is around 2.5, and then the FADH2 is around 1.5 ATP. So then we have our pyruvate dehydrogenase complex and our citric acid cycle. So our citric acid cycle is actually going to help us to generate even more ATP. So when we go through this entire process between glycolysis and the Krebs cycle, we're going to generate even more ATP. So we see that we have a lot of intermediates in the citric acid cycle. Those actually can be formed from other places as well. So for one of them, we can actually get our pyruvate from amino acids, which we'll talk about where we get those in just a minute. But those can be derived from amino acids in the form of pyruvate. And then another important intermediate is oxaloacetate, which is derived from aspartate. And then alpha-ketoglutarate is a huge intermediate that can be derived from glutamate. So we have these that we can see come from our amino acids that are intermediates in our citric acid cycle. So in our citric acid cycle, we are going to form NADH and FADH2. So these are really important because these add into our electron transport chain, which is going to be our real mechanism of generating lots of ATP. So that actually does so through the electron transport chain through proton pumps. So we have our NADH and our FADH2, which donate electrons to kind of get the energy flowing. And then we create a proton pump. And so in order to do this, we're going to pump protons from inside of the mitochondrial matrix to the inner membrane space. And so this one over here has a better look at our proton pumps. So this is actually called ATPase. So it's going to actually, with the addition of ADP and a PI, a phosphate, we know that those can make them an ATP, right? So how does that happen? In our ATPase, there's going to be a conformational change that's triggered by the addition of that ADP in the phosphate. and then the conformation will change, is gonna put out a water molecule and some ATP. So that's really interesting. That's how we can generate a lot of ATP, and between those three processes, we'll get around 30 ATP. So some people say between 30 and 38 because it's constantly going and it's kind of hard to quantify. So something we were talking about earlier was the amino acids that can contribute to our intermediates. And so those actually can come from things in our diet as well, or things that we intake as well. So try to stand up. So over here we have our CO2. So this is our other starting point that we were talking about. So we have CO2, which is going to be turned into carbamic acid, and then so we're going to use PRP and lots of other chemicals, and we're going to end up with a enosinate. Okay, and then I'm just going to go through several other mechanisms. We can also use our glycerol 3-phosphate, which came from glycolysis, which was an intermediate in glycolysis. So we can use that to form triglycerols, and then even cholesterols down here. But let's look at the amino acids. That's the really important one for the ATP synthesis. So I was talking about how we form enosinate from ingesting carbon dioxide. And so that's our starting material here. We're going to be able to form UTP, CTP, TTP, ATP, and GTP. And these can go on to help form our amino acids. Glutamate and acetyl-CoA can form arginine. 3-phosphoglycerate can form serine, glycine, phenylalanine, tyrosine, thymine, cysteine, and so acetyl-CoA is actually going to be a really important cofactor in all of that. So anyways, that's the formation of our amino acids, which can go on to form our glycolysis Krebs cycle intermediates. So that is that. Oh, and one other thing is that in the formation of these amino acids in our cholesterol, this is actually a couple of the steps are going to take ATP to be able to produce. So one of them is, so like our NADPH, that's an intermediate from our other cycles. There's one. Oh, so like 5-pyrophosphate melanate into to give our CO2 and a phosphate is going to take an ATP. So that's how we see that we need to be able to do these pathways and be able to form these, and we need these pathways to form intermediates for these, and it's a big cycle.